Hi everyone, I hope you're doing well and staying safe. My name is Lars Lindgren and I am graduating this May with degrees in cell and molecular biology and honors arts and letters. I have finished my honors thesis and it's titled Co-overexpression of AtCLCc and PP2AC5 to increase salt tolerance in Arabidopsis thaliana. So it's a little bit of a mouthful, but what it really means is that we wanted to find a way that we can improve the ability of crops to withstand abiotic stresses. These stresses include salt, drought, and heat, and my project focuses primarily on salt. And the reason for this is because every single year there is a reduction in viable farmland that can be used for growing crops. This is a result of several different things. Just a few of them include over-farming, over-fertilization, and even urbanization. And at the same time, however, there is actually an increase in the world population. More and more people are being born every year, which means that there's more and more people to feed and more people to provide clothing for. And of course, this creates a really difficult problem for us to solve. How can we take care of more people with less agricultural room to work with? One way we've taken to approach this problem is by genetically manipulating crops to better withstand stresses like salt. So to do this, we've taken two genes, AtCLCc and PP2AC5, and co-overexpressed them in Arabidopsis thaliana. So Arabidopsis itself isn't really a practical crop. It's mostly used as a model organism in plant biology, and that's because it grows really quickly and gives out a lot of seeds. It can be relatively easily manipulated. However, it can also be used to provide a proof of concept for a genetic construct that can then be incorporated into more practical crops like cotton. So what we've done is that we've taken AtCLCc and PP2AC5 and overexpressed them in Arabidopsis. AtCLCc is actually a antiporter that rests on the vacuolar membrane of the plant that takes chloride ions and moves them from the cytosol to the vacuole. And so when chloride reaches high enough levels in the cytosol, it can actually create cytotoxic effects that damage and inhibit a plant's growth. So we can actually minimize these effects by moving the chloride into the vacuole. PP2AC5 achieves a similar goal, but in a slightly different way. It actually is a gene that upregulates the chloride channels in a cell, and these channels include AtCLCc. So not only do we have more of these chloride channels are actually more active and more efficient. And so after we've done this, we've tested these plants on different growth plates with different varying concentrations of salt, and we were able to actually get, while not significant, promising data that allows us to further our research into these two genes and go beyond what we've done thus far. I have really enjoyed my time on working on this thesis because it's really given me something to take ownership of and really round out my education and figure out how to work on a huge project like this. I joined my lab 3 years ago now in the summer after my freshman year, and so it's been a really nice way to kind of taken everything I've learned up to this point and put it into one project. I want to thank my family and my friends, and of course my PI and thesis director, Dr. Zhang, for his mentorship, and my lab mentor and second reader, Ruvini, for everything she's done and for helping me as I've gone on this amazing journey. So I want to thank you all again for watching, and I hope you all have a great day.